Welcome to practical demonstration. Here are the laboratory apparatus and materials that we use for experiment for cell division or mitosis. Compound light microscope, measuring cylinder, beaker, filter paper, watch glass, clean slide and cover slip, test tube holder, white tile, blade, mounted needle, water bath, prepared slide of various stages of mitosis, ice cold water, onion root tips, hydrochloric acid, aceto orcein, and acetic alcohol. Exercise 4.1 Prepared slide of mitosis. Step 1. Examine the prepared slides of various stages of mitosis. Step 2. Draw and label the stages of mitosis observed. Exercise 4.2 Preparation of onion root tip slide. Step 1. Rest onion bulb on the rim of a container of water to allow the roots to develop for 3 to 4 days. Step 2. Measure the root tips for about 1 to 2 cm long. Cut off the root tips. Add a small volume of acetic alcohol on the watch glass. Then, transfer the root tips into the acetic alcohol and leave it for 10 minutes. Step 3. Wash the root tips in ice cold water for 5 minutes. Transfer the root tips from ice cold water onto filter paper and let them dry. Step 4. Measure 3 ml of hydrochloric acid. Transfer the hydrochloric acid into an empty beaker. Put the beaker in the water bath for 5 to 10 minutes. Maintain the temperature at 60 degrees Celsius. Transfer the root tips to preheated hydrochloric acid and leave it for 5 minutes. Repeat step number 3, but be cautioned, the root tips will be very fragile. Step 5. Transfer the root tips onto a clean microscope slide. Measure 1 mm from the growing tip of each root tip. Cut each root tip. Keep the tips and discard the rest. Step 6. Tease the root tips with a mounted needle. Add 1 small drop of aceto orcein on the root tips. Leave the stain for 2 minutes. Step 7. Cover the root tips with a cover slip. Blot firmly with several layers of filter paper. Press gently to spread the root tips. Step 8. View the slide under microscope using high magnification objective lens 40 times. Handle the microscope as what we have learned in experiment 1. Observe the chromosomal behavior in the mitotic stages. Step 9. Identify, draw, and label cells showing the stages of mitosis.